such a nice day. The weather's always like this in Belfast, obviously. So it's a great pleasure to welcome you to this meeting on livestock, companion animals and wildlife coronaviruses. This is one in a series of meetings that is organised through the UK International Coronavirus Network, which Sharon will tell you a bit more about in a moment. We're very fortunate to have a number of sponsors, very generous ones. So I just want to mention those. Obviously the UK ICN itself, SIVA, and we also have some sponsored lectures by the Microbiology Society in the UK and the Royal Society of Biology as well. So I'm going to hand over to Sharon to tell you a little bit about the network.
Thanks, Louise. And yes, welcome everybody. I'm sorry so many of you have had troubles traveling here, and we have a few colleagues who will be a little bit delayed this morning thanks to BA Airlines, but never mind, they will be here. So yes, I'd like to welcome you all on behalf of the International Coronavirus Network, whose director is Julian Hiscox from Liverpool University. I'm one of the co-directors, and Dallin Bailey, who will be joining us later on this morning, is also one of the co-directors, and we've got Lisa and Jill here from our Secretariat and Administration, so thanks very much to them. I'd like to say thanks to Louise and Mia for coordinating the meeting here in Belfast today. The International Coronavirus Network was set up a couple of years ago through sponsorship from DEFRA, which is the government organisation responsible for food farming and the environment here in the UK, and also from BBSRC, which is one of the research councils that sponsor academic research here in the UK. And we are very pleased to have SIVA on board to sponsor this meeting as well. So it's a really nice mix of government institutions, academic institutions, commercial interests that has brought us all here today. And one of our key targets is not only to have the presentations on the three topics that Louise has introduced, but to also run a workshop tomorrow afternoon. And that outcome of that workshop will contribute to STAR IDAS, which is being hosted by Valeria, who'll be here later on this morning as well. And the outcome of that will be a gap analysis and a roadmap on coronaviruses in these three topics, and will be used to inform funding organisations for future research in this area. Pardon me, and I'd also like to say thank you to Cyril Gay from USDA, who's come to be part of that workshop, and will also use part of the outcome of the workshops in the afternoon to educate the process over in the Americas. Thanks very much, and thank you to all of our invited speakers and the offered papers who you're going to hear today. And I'll hand over to Louise to introduce our first speaker. Thank you.
So I'm very pleased to introduce our first keynote speaker, Professor Elmer Chillian from the Pirbright Institute. And this lecture is actually sponsored by the Royal Society of Biology. And Elmer's going to talk about porcine respiratory coronavirus as a model for acute respiratory coronavirus disease. Thanks, Elmer.
Thank you very much for the introduction and the invitation. It's a real pleasure to be here in these glorious surroundings, and I'll share our work in progress on the pig coronavirus model. So, as you all know, coronaviruses are widely distributed in mammals and birds. In some species, they are detected as a result of surveillance programs, and we don't know the burden on the host, but in most species, they cause respiratory diseases or gastrointestinal diseases or diseases on the central nervous system. And they also affect animal welfare and of economically important species. And respiratory viruses, as we know, are a huge global health problem. And the coronaviruses were responsible for 3 major human epidemics, including the latest pandemic. And in all of these cases, the emergence of coronavirus in humans was associated with zoonotic transmission from animals. And as I showed on the previous slide, many livestock species, including pigs, are infected with coronaviruses, which leads to very substantial economic losses. And I'm going to talk about porcine respiratory coronaviruses, which are globally endemic in pigs. And we need to understand much better the fundamental biology of the pig coronaviruses, because there are recent reports for a zoonotic transmission to humans. So, porcine coronaviruses. I'll start with the transmissible gastroenteritis enteric virus, TGEV, which was identified in 1946. And this is an alpha coronavirus, and it causes acute diarrhea, vomiting, dehydration, and young piglets can die after 5 to 7 days. In 1984, the porcine respiratory coronavirus was identified in Belgium, and it evolved from the TGEV, and there was a deletion of about 200 amino acids in the spike of the TGEV, plus there were also some deletions in the accessory 3 genes, which changed completely the tropism, and this virus is entirely respiratory. It induces a very mild or subclinical disease, and there is always pneumonia, but the disease can be exacerbated if there are secondary infections. So I think, and what happened is that PRCV is globally endemic in the pig herds, and that provided cross-protective immunity against TGEV, and therefore the burden and threat of TGEV is significantly reduced. And perhaps we all can hope that the same will happen with SARS-CoV-2, that such a variant can evolve, which can provide cross-protective immunity to other more pathogenic strains. However, the good story ends here with the porcine coronaviruses, because there are a lot of emerging and 3 emerging coronaviruses which pose a huge economic burden. Porcine epidemic diarrhea virus, the economic losses in the US are calculated between 900 million to 1.8 billion. There are recombinant strains identified between TGEV and PEDV, porcine hemagglutinating encephalitis virus, and the swine acute diarrhea syndrome coronavirus was identified in 2016, I think, in China. And again, it's a very serious, can cause serious disease. And also porcine diarrhea coronavirus was identified in 2012, again in young piglets in Hong Kong. And what is worrying about these 2 viruses is for the swine acute diarrhea syndrome coronavirus, it was shown that it can replicate in primary human cells from the lung and intestine, and therefore there is a possibility for a zoonotic jump, and also it evolved from a from a bat coronavirus, this virus. And also there was a report in 2021 for that 3 children from Haiti were identified with undefined febrile illness, but in all of them, porcine Delta coronavirus was identified. So really we need to understand better the pathogenesis and immune control of these viruses. So back to the porcine respiratory coronaviruses, why we are interested in that is that Linda Saif team, after the first SARS epidemic in 2002, she published a paper in 2007 where they described that the pathology which was induced by PRCV strain was very similar to the pathology observed in, and the pneumonia observed in SARS patients. So they used this model to assess the corticosteroid treatment in this model. But we were very interested in this study, and we were very keen to reproduce and establish the model because we had a lot of experience in exploiting the pig as a model for influenza. And the huge advantages and what makes the pig an excellent model for influenza is that it is a natural large animal model. It is infected by the same subtypes of viruses which infect humans, and especially we work on H1N1 pandemic 09 virus, which is antigenically similar between humans and pigs. And also immunologically, physiologically, and anatomically, the pigs are more similar to humans. Very often we perform studies head-to-head in mice, ferrets, and pigs, and we see a lot of differences. For example, vaccines which protect mice and ferrets do not protect pigs. And I guess that they give us a rosier picture than the pigs. And also the tracheobronchial structure, the airway morphology, the electrolyte transport are very similar between the pig and the human. And also when we were working on the pig model to study influenza, we developed a lot of tools to assess the porcine immune responses. We have inbred Abraham pigs, they have identical MHC, which allows to use tetramers to define the fine specificity of the response. We have done in-depth analysis of the responses in influenza in pigs. We also can assess tissue-resident memory cells. These are the cells which are incredibly important for immunity against respiratory viruses, and you can do that by infusing intravenously antibody, which labels all of the circulating cells, and the tissue-resident memory cells are not labeled. So that is very easily done in mice, but in pigs is very, very challenging. But we have done that. Also, we can perform scintigraphy, so that is scintigraphy in pigs with, using technetium, so we can follow and assess the distribution of particles in the respiratory tract, especially if you administer vaccines or therapeutics to the respiratory tract. And also, unlike ferrets, for example, which are the golden model, we have a lot of reagents to do a very high dimensional phenotyping of the porcine lymphocytes. We can identify all major lymphocytes and leukocyte subsets, and that is also a figure showing that also we can perform single-cell RNA-seq. So basically, we wanted to transfer all of the expertise and knowledge which we had from the pig influenza model to the porcine PRCV model. So what we did is, back to PRCV, we wanted to test what Linda Saif reported. She used the strain RSU1 to infect pigs. So we also infected pigs, that was a pilot study, with other strains which we found in our freezers. And all of these strains had similar growth characteristics when assessed on swine testis cells or porcine kidney cells. But when we infected the pigs, they induced a very different pathology. So the pathology was assessed by these consolidations. So ISU1 induced quite a mild lung pathology as reported by Linda Saif's team, while, for example, 135 induced quite much more severe pathology compared to ISU1. That was also confirmed by the histopathology. As you see, there are a lot of macrophage lymphocytes and neutrophils infiltrates, and that is the staining for nucleocapsid protein, and there is more infiltration and histopathology damage in the 135 infected animals compared to the ISU1. Also, the pathology the model was very good to assess the pathology of the upper respiratory tract. That was analysis done by Fabian Lin at APHA. So he detected virus antigen on the olfactory as tentacular cells. So you know that in COVID there was reports that the virus can affect smell, and we can detect the virus in the nasal mucosa, but we cannot detect it in the nerve, the olfactory nerve. We also assessed the replication of the PRCV in the respiratory tract. So there was replication in the nasal swabs. There was obviously shedding from the virus in the nasal mucosa. And what was interesting is that the viruses which were inducing class pathology had replicated less in the trachea and in the lung. And we were very pleased because we also set up in vitro tracheal organ cultures and also in vitro primary cells, that this was replicated in these in vitro cultures, which can facilitate to assess in much more depth the very early events and pathogenesis of these viruses. We also established in vitro lymph node organ cultures to study adaptive immunity. So that was based on a paper which was by Mike Davis in California. So that was really a remarkable study where they took tonsils from humans and they disaggregated the tonsils and they put them in these transwell cultures. But what they did is they stimulated the tonsil with live attenuated influenza virus, and they claimed that they managed to see an antibody response in B-cell differentiation in vitro. So after 7 days in this culture, they were able to see antibody response and all of the events which happened during B-cell differentiation. So we set up these cultures, but we did not use tonsils. We used the tracheobronchial lymph nodes. And these are, and put them on these transwell plates. And indeed, the cells from 135 infected animals, when they were stimulated in vitro with 135 virus, after 7 days, we can detect a considerable antibody specific for the spike, and also this antibody also neutralized. So in our case, it's much easier to reproduce that, while here, you don't know when the humans were exposed to flu, but here we consistently see a very good comparison, and we can reproduce the adaptive immune response in these cultures. Or to summarize this part of the story, so far we established the pig natural health coronavirus model. There are obviously different strains which induce different degree of lung pathology. The trachea and the lung cultures can predict pathogenicity in vivo, and the in vitro organ cultures reproduce the antibody response. So then we said, can we use this model to ask the fundamental questions? And we were interested in these questions. What are the key events that determine whether a virus causes severe or mild disease? What are the immune mediators of severe and mild pathology? And can we use the model to test novel therapeutics and vaccines? And that is very much work in progress, so I'm going to show unpublished data, so that is, the studies are not complete at all. So to answer these questions, what we did is we infected pigs with ISU1 or 135. We chose these two viruses, and we performed staged postmortems. So we caught pigs on day 1, day 5. Day 1 because the very early time points are the most important if you want to determine what happens. At day 5, we have the best pathology. Day 11 is the peak of the immune response, and at day 20 now you can probably go into the memory response. So we assessed the shedding of the virus. There was shedding, there was more shedding from the 135 infected animals, and as before, we assessed the virus load into the trachea, bowel, and lung. As before, there is more replication of the 135 virus in the trachea, bowel, and lung. After day 11, we cannot detect virus. We also assessed the antibody responses. Again, the 135 infected animals showed a much higher antibody response. We assessed this by ELISA against the recombinant spike, and that was the case for the serum IgG BAL, that is the bronchoalveolar lavage, so that is the lung wash, and in the nasal swabs, and also the same was true for the IgA responses. We also performed neutralization assays, and what was clear is that animals infected with one virus can cross-neutralize all of the PRCV strains. So these are the responses. So animal infected with ISU1 in black, 135 in red. That is cross-neutralization against all of the strains we have, including TGEV. We also assess the T-cell responses, and that is flow cytometry on the CD4 cells from the lung wash. And again, the 135 animals show a much stronger response. Here is secretion of interferon gamma and TNF, and that is very similar to humans. There are a lot of assays in humans where there was shown that there is a very strong CD4 response in COVID patients. And also we assessed obviously the CD8 BAL response, and again, there is a much stronger response in the 135 infected animals. We are also assessing the fine specificity of the T-cell responses. So we infected also inbred Abraham pigs, which have the same MHC. In pigs, it's called swine leukocyte antigen. And we identify epitopes that is in the nuclear capsid, that is in the spike. These are CD8 epitopes, that is CD4 epitopes, that is irrelevant control with flu peptides. But why that is important is because when we analyze the responses in the pigs against flu, what we saw that there is a very different specificity and hierarchy of the responses in the respiratory tract compared to the blood. And so that is very important for clinical trials where humans are only sampled in the blood, and you do not have a correct picture of actually of the what are the important responses. They are very different between the two sites. We also are doing a very extensive phenotyping of cells. That is just an example of the phenotyping of the CD4 T cells in pigs. They express CD4 and also they express CD8 alpha, the T cells in the pigs. And we, based on this staining, we can differentiate naive effector memory, central memory, and terminally differentiated cells. So, and we are doing that with all of the major leukocytes. And I'll stop here about this model, and I'll go now how we use the pigs as a model to study immunogenicity of the COVID vaccines. So we can use this PRCV model, and we are developing at the moment that to assess vaccines and therapeutics. We can assess the efficacy of these vaccines and therapeutics in the PRCV model, but then you have to have in your vaccine obviously the pig antigen. So, but that is in development. I'm not going to show that, but what I'll show you is how we use the pig as a model to test the vaccine immunogenicity of the COVID vaccine. So that is just plain pigs that are not infected with PRCV. Pigs cannot be infected by SARS-CoV-2, that was shown, but we, as I alluded to before, we use the pigs as a model to assess the immunogenicity of vaccines for flu, and we show that it's a very, very reliable and excellent model. Anyway, so that happens in 2019, and that was a really huge collaborative effort, and we tested a lot of vaccines. These studies were led by Simon Graham. And I'll show data only of how we tested the ChAdOx vaccine, and that is another candidate vaccine. So, so that is 3 years back. So at that time, what we knew is that that is a replication deficient chimpanzee adenovirus. It was expressing the full length of the spike protein, and at that time, all we knew is that a single dose protected macaques against disease, and it was incredibly important to assess whether there will be a boosting effect of a second dose of the vaccine. And what we did is we immunized pigs with chow dogs. In red are single dose, in blue is prime boost. And as you see, there is a very strong boosting response. That is the interferon gamma ELISpot that assesses the ability of the T cells to be stimulated and to secrete interferon gamma. And here is a flow cytometry analysis where you dissect whether it's the CD4 or the CD8 T cells which secrete these cytokines. So, and that was in contrast to the mouse study. So in the mouse, there was no boosting effect. And also, we assessed the antibody responses, and again, there was a very strong boosting effect. There was ELISA with the full-length spike, that is ELISA with the receptor binding domain, that is pseudo-neutralization assays, and that is the virus neutralization assay. And these titers were comparable to the titers from convalescent patients. And so based on this study, they informed the introduction of a booster vaccine because, again, in mice, boosting of the antibody response was seen only in the inbred bulb C mice, but not when that was done in the outbred strain. Well, as I said, in the pigs, we saw a very strong boosting response, and that after that was reproduced also in humans. So the pig was quite important in informing this decision. And that is another vaccine which is now in development. That is a spike protein receptor binding domain, the RBD, which is delivered on a virus particle using tag spy catcher technology. That is like a glue which glues the antigen to the virus particle, and we assessed different doses of these RBD spike VLPs and also compared them with the spike protein given with an adjuvant. And in this case, the VLP did not induce any T cell responses, so the T cell responses are not at all impressive. However, they induce a very strong antibody response, and there was a very strong boosting effect. And this vaccine now is in clinical development. So that is just to illustrate the power of the model to assess immunogenicity. There is my concluding slide that we established a large natural host animal PRCV model that ISU1 and 135 PRCV induced different magnitudes of immune responses which correlate with severity of lung pathology. At this stage, as I said, that is very much work in progress. We do not know whether it's the virus or whether it's the immune response. What is clear is, however, as in humans, that you have to control the excessive inflammation and to control disease. What also is clear from our studies that the in vitro tracheal and lymph node organ cultures can predict events in vivo, and that can really facilitate to dissect the molecular events which are happening, and that the pigs were an appropriate model to test vaccine immunogenicity, and that I hope I convinced you that the pig is a powerful model to investigate strategy for prevention or amelioration of coronavirus infection and disease. And this work wouldn't have been possible without the efforts of many, many people. Here I did not put on the slide the groups which were involved in the testing of the COVID vaccines. I just acknowledge here Sarah and Teresa, who developed the Chadox vaccine, and Alan and Jack and Pramila, who developed the spike tag VLPs. And in Purbright, Sarah and Veronica set up the model. I showed data from Isabella and Eshan. Simon led the vaccine studies, and Fabian and Alex at APHA performed the pathology, and the animal studies were also performed at APHA. And thank you very much.
Thank you very much, Elma, for a really fascinating and interesting overview of the porcine coronaviruses. Would anybody, any questions for Elma? Yeah.
Hey, I'm Steve Reckett from the U.S. Department of Agriculture. I'm wondering if you know, out in the wild, if they ever record.
Yeah, sorry. Yes, everybody, the conference is being recorded, so if you want to speak, please wait for the microphone.
Start over. Is this
Shout.
one working? Okay. I don't know. I'm Steve Reckett from the U.S. Department of Agriculture. And I wonder, out in the wild, if the different viruses, how they interact, if it's more the ISU strain or the 135, if both are in a herd, if one is going to predominate, and kind of what happens in the herds.
Yeah, a very good question. I don't know the answer. At least in Europe, or at least in this country, we do not have surveillance for porcine coronaviruses. But very recently, I performed an experiment with coronavirus, which was isolated from Belgium, and it was from a herd in Belgium. And this virus, which obviously induced a very mild subclinical disease in the pigs, induced a very severe mild pathology as the 135 strain. So they're out there, but as far as I know, there is no surveillance.
Thanks.
Thank you. I guess models are all about consistency. You want a nice, consistent response, which makes sense. But one of the things about COVID, obviously, is variation in clinical signs, age, for example, but also in vaccine. You know, we all hear these stories of people responding badly to vaccines, and that's one of the issues that has affected vaccine uptake. So I wonder if I guess my question is, in a model system like this, can you look at variation in response as well to clinical disease and to vaccines?
Yeah, that's why we are using both outbred and inbred pigs. So we have a variation in the clinical presentation, as you see. Actually, they do not show a lot of clinical signs. So it is a very mild disease, although we give quite a lot of virus. We infect with between 1 by 10 to the 7, 1 by 10 to the 8. That's why we are using outbred and inbred pigs, but there is variation. And so far, that's why we set up the model, because you remember all of the stories that vaccines can increase pathology, et cetera, and we have not tested that at the model, but there were no vaccines which can increase pathology so far as I know. But it's a very valid point. Yeah.
So I guess the obvious variation is age.
Age,
COVID.
I know.
And I guess you use piglets.
No, these are 6 to 8 weeks old. We have performed it also on 3 to 6 months old pigs. But don't forget that pigs put on weight about 5 kilograms per week. So yeah, it's very challenging to do the studies in an aged pig.
But you might get a massively different response in an adult pig.
Yeah, correct, yeah. But that will be prohibitively expensive, yeah.
Can I just ask Elma about the Delta coronavirus in the children? I mean, has that occurred before, or what are the receptors involved?
That was, there was a lot of genomic analysis, and I'm not a virologist, and but they were showing that there are some deletions and adaptation of the spike to change the ability to bind to the human receptor. So there is a lot of that in the paper. That is a Nature paper. Yeah.
Okay, so another one we might need
Yeah.
to worry about. Okay, thanks very much,
Thank you.
Elma. Can we just thank Elma again?
Thanks very much, Elma, and I'm sure there are plenty of other questions and people can ask those when we get to coffee break. Okay, I would like to invite the first of our offered papers, and that's Patrick Dunlop from Ulster University. something's in the air, Patrick. Thank you.
Yes, thanks very much. Good morning, folks. I'm a mere engineer, School of Engineering at Ulster University, but during the pandemic, as the previous speaker described, I think we were all keen to try to support, to help, to see what we could do, to develop technology or to consider different applications that might be useful during that time. So I'm very lucky to work with some great microbiologists at Ulster, here on the slide and in the audience with me today. And I also knew some brilliant virologists at AFBI, who, as part of our multidisciplinary team, we tried to see if we could identify a technology to reduce what might be airborne bioaerosols. I have a company down in the bottom right-hand side that contacted us during that time, who wanted to develop technology along this field. So whilst Illumex still make and sell air treatment systems, they were involved in providing the technology that we used. They don't sponsor the research. We're not in any way connected with them. We sort of test their boxes on their behalf. And their support is gratefully appreciated as well. So something in the air. If we look at this paper co-authored by Chief Medical Officer from England very recently in Nature, they describe a number of different pollutants that might be in indoor air. Indoor air has all of a sudden become the new challenge. How do we disinfect? How do we identify what might be present in indoor air? But that paper still highlights problems with perhaps smoke, fire, cooking, pollutants from the back of diesel engines, etc., etc. And only at the very end of the paper do they begin to mention and describe bioaerosols and how there could be a risk of transmission of a number of different pathogens, including coronaviruses, through transmission with that airborne route. World Health Organization during the SARS-CoV-2 pandemic were very quick to rule that out. But I think maybe as there's been a little bit more education, and if I go back to my coughs and sneezes, spread diseases, respiratory sort of ideas, there's a larger body of evidence now that suggests that there are airborne bioaerosols, and that is an effective route of transmission. Not only in humans, and perhaps what I guarantee you are a more educated audience than I am, there could be applications then in animal husbandry, where we've got very highly densely populated systems, food production systems that are critically important to veterinary and human health as well. And we heard a little bit about that in the first talk. So something in the air, whilst we can perhaps see some of these things, we absolutely know now there are things that we can't see, and that we need to begin to develop technology to have safer environments, both for humans and for animal welfare as well. So the aim and our approach for this project was, of course, there are 101 different systems that you could use. Most of those filtration systems, use filters. They take pathogens, they pass them through, akin to a face mask that we might use. They trap them on the surface and effectively build up quite a large reservoir of pretty nasty things on the surface of a box that might not be looked at for multiple months, maybe years. So our approach was to try to sterilize the air. We wanted to achieve something akin to a 6-log reduction in pathogens from the entrance to the exit. We were pretty set that we didn't want to use filtration-based systems. They collect dirt, they collect dust, they collect pathogens, and have to be maintained. So we wanted, as easy as possible, a pathway for the air to pass through the systems so we could treat larger volumes at lower cost and higher throughput. We were also interested in a scalable technology. The air in this room versus a large poultry house versus a medical ward, all of different sizes and shapes, and how could we have a technology that could be scaled and could be applicable to a wide range of applications. I suppose luckily in this case, or unfortunately for me, myself and some colleagues had about 20 years experience of working in water disinfection. So we were very familiar with the pros and cons of UVC inactivation. We are aware, and people are very familiar with that technology as a proven biocide. During the pandemic, There were many other technologies that were touted as being effective, which still haven't been proven, just put it like that. So along with our laboratories and expertise and the partnership with Illumex, we worked to try and develop a UVC disinfection system that could sterilize air. Coronavirus, it turns out, is quite a sensitive little object. So we used a number of different surrogates to help us with the development of technology. We didn't have access. I don't think very many had access to virology labs and the ability to test with SARS-CoV-2 during that time. So we began to work with a number of marker molecules. So effectively, this little schematic shows the box that we developed to include our UVC systems. We were able to introduce bioaerosols then in through, and they would maybe the wee animations aren't working on this one. They were able to pass through the system to be exposed then to a number of different UV systems, UV sources that were within the box. A first lamp, a second lamp, third lamp, fourth lamp, and fifth lamp. So the bioaerosols could pass through this longer path. Effectively, we had a 3-meter path length in a 1-meter box as a result. We were able to mix the air as it went through. We were able to light each of these lights in sequence. We could study the effect of the power of those lamps. And the exposure time as our air passed through was less than 1 second. So we were trying, as I said, get 6-log reduction of pathogens with 1 second of treatment with no loss in pressure across the system. Okay, so there wasn't a pressurized system in any way, shape, or form. And one of the ways that, from our physics background, we wanted to understand was if we had reflective liners inside the walls of the chamber, how could we maintain the UV energy within the system for longer rather than have it effectively leave the system more quickly? So the UV energy effectively bounces through the system. Okay, our aluminium filter acts like a standard mirror. It bounces once. But with our PTFA's Teflon, perforated Teflon, it's a little circle, pores of materials. The light scatters in a totally different direction. Some of it scatters back, some of it scatters through the system, and you effectively have more power per unit space with that material based upon its reflective properties. So if the little animations were working, you would see this little bioaerosol that passes through the system and back out. We generated, I bet the videos weren't working either. Oh dear, dear, dear. All that time to make the videos. Never mind. This over here shows a little atomizer. So we're able to take liquid in which we have placed, for example, a PBS, including our different pathogens then, or in case of coronavirus, then it was a tissue culture media. So we have a liquid that we can make up in our standard laboratory processes. And again, so we take that liquid, we drive air through, and it atomizes it into a bioaerosol with a particle diameter of approximately 1 micron. Okay, so there's a distribution of sizes of those little particles, but on average, particle size is 1 micron. So that's very akin to what might be the size of particles from a cough or a sneeze. Those act and come down this little chamber into the system as like a fine mist. And a bit like the smoke you saw in the first picture where you could see something in the air. If the light is right and the video worked, you would see all of these little aerosols being brought into our treatment chamber. As I say, they pass through and we take them back out then. We sample the air at this outlet port. We don't collect all the air. We sample at a constant rate. We draw air out and we put it through what is like a little cyclone system here. The little cyclone spins the air through it. And any particles that are trapped, that are in the air, are transferred back into that liquid phase. We simply then switch off the pump, take that liquid, and go into your standard microbiology or virology processes with that liquid phase. So we, as I said, didn't have access initially to the coronavirus samples, so we worked with some model organisms. A model bacterial pathogen, Staphylococcus aureus, but a model virus, MS2. It's actually a very UV resistant virus that we use for our water studies, and it grows in an E. coli host. So in a standard lab, as such, we were able to do a lot of this work. Along the bottom axis here, we see the intensity or the exposure, the UV dose, the power of those lamps within the system. And you can see if we've got 1 lamp, 2 lamps, or 3 lamps, obviously we're going to increase in the power within the system. But also we see an increase then from the black dot to the open dot, from the aluminium to the PTFE, where we have an increase in the power of our system based upon simply that reflection on the wall. As a function of time then we've got log kill, our survival fraction on the y-axis. We see a very significant kill right up until, well, for our MS2, the most that we could have in the system was 6.5 log, and we were able to reduce that down as low as our detection was, so a complete inactivation of that virus. What I should say is that initially we would run the system with only air, with no UV lamps. So if there was any loss of virus as it passed through the system, we were always measuring against that air-only baseline and measuring the effect then of the lamps as I show on the screen. So a very effective kill, and we could study that then as a function of dose and go back and tweak the UV lamps we would use in this system in terms of scalability, if we can understand that dose response. Staphylococcus aureus give a very similar response. We see that increase as a function of a reflection. But the nice thing here was if we can do something simple with bacteria as a surrogate on a model, it saves us having the complexity of the virology. So we knew we were onto something that worked, and knowing Louise and knowing some of the other staff in AFB, it was like, would you do just a little experiment here and see if we can kill some coronavirus? And they were very, very generously offered to host us for a few days to undertake some studies, which I guess the audience are much more familiar with than me. So we see here we ran the same idea of an experiment where we had bovine coronavirus then in our bioaerosols that passed through the system, and we investigated the effect by tissue culture. So hopefully these have come out a little bit so you can see. Up at the top, we have a tissue culture negative control where we get a confluent growth of cells across the surface. If we induce a cytopathic effect, we kill those cells, we see they begin to float. Dead easy for most of you. If we run our system through coronavirus with air only, so no UVC system, the coronavirus is still viable, and it gives this sort of effect where it kills the cells. When we come to the lowest level of power in our system and we run our coronavirus through, we go back and we see there's no cells are killed here. The coronavirus is inactivated, the tissue culture cells are still alive. So we're able then to measure and quantify that effect using TCAD50 calculations and try and understand our log kill of our bovine coronavirus through the system. As I mentioned, coronavirus is actually fairly physically delicate. So although we would have had a higher titer than that to begin with at the start, there was some damage initially mechanically as the coronavirus passed its way through the system. But even with the smallest or lowest dose of energy, we were able to totally inactivate the pathogens within the bioaerosol. So I was interested in, okay, if there's something in the air and if it's inactivated, can we demonstrate that it's still physically there? Because unlike our filters, we don't, in UVC systems, remove those pathogens from the air. They're inactivated, as we showed previously, but are they still physically there? And the guys in the lab had this idea that maybe we could use a bit of RT-PCR to go and look and see, is that biological material still present? It's just not capable of causing infection. And if we look as a function of the dose then, we still have a very consistent CT value, representative then perhaps of the same concentration of that virus being present in the air. So whilst there remains something there, it's no longer capable of causing infection, which was our primary goal. So finally, to conclude then, I think we've shown that UVC disinfection can give high levels in a single pass. So most filtration systems require multiple passes of the air through the box. We can do that in a single go. What was also nice was that we were able to show that we could use some of these model pathogens to demonstrate the efficacy against a coronavirus. During that time, that was a huge interest to the commercial companies to say that their systems had been tested and did demonstrate efficacy. There's another video not working there, which I hadn't bothered. As a lesson to anybody, don't try the videos. We were able to then do a lot of mathematical modeling to try to work out how we could scale the systems. And that's led us now to work with some poultry farmers across in England to investigate the potential then to disinfect air coming into the houses to perhaps prevent the risk of avian flu. I'm delighted, as I said, to be here, and thanks to all the collaborators that I mentioned at the start. I'll be about for the rest of the day, and I suppose my purpose here is if people are interested, it's really to try and see how we could use this technology to the advantage of those that are in the audience. And that's me. Thanks very much.
Great. Thanks, Patrick. Are there any questions? If not, while people are thinking about it, I can ask one. No, that's fine. So my question is, with your model systems, you said you use PBS and tissue culture media as the kind of delivery media into your experimental system. So I know from studies that we've been involved in with UK HSA from a public health perspective that having respiratory excretions makes a huge difference to virus survival in a lot of these scenarios. So just kind of whether you could mention what components, so I guess the tissue culture media might have had fetal calf serum or something like that in, but probably nothing as complex as a respiratory secretion to protect the virus in any way.
We would have had, I suppose the tissue culture media was quite sticky and gloopy. I can tell you that from what was left on the inside of the system. And there may be that protective effect. The reason that we wanted to use such high power of UVC was exactly for that reason. I guess if we can work with the bacteria, which are much larger physical structures and still cause inactivation, we would expect those more complicated systems to hopefully give some level of efficacy and give some level of confidence that if there is a more complicated biological media, that the UV can still penetrate through that and inactivate the DNA or the RNA.
Okay, great. Thanks. Any other questions for Patrick? Elma?
How would you comment based on the recent studies that actually aerosol transmission was not so prevalent? I think there were quite a lot of papers recently showing that actually it is much more direct contact rather than...
So I guess there can be a number of routes of transmission. And I know some studies we've done in the hospitals where there's been evidence to show that there has been airborne transmission and they can actually follow infections through the ward based on how the air moves. So I guess it's a complex problem, complex disease, complex routes of transmission. But I think there is a body of evidence that does show airborne does play a role. It's not the only, absolutely, not the only mechanism or means of transport, but that there is a role to be played there for looking for indoor air hygiene in general from a number of different threats.
Thank you.
Thanks.
We're now moving over to our next speaker, who is Carlos López-Figueroa from the Centre de Recerca en Sanitat Animal. Thank you, Carlos.
Okay, good morning. I'm Carlos. I'm a student in IRTA-CReSA. It's a center of research in animal health, and we study different species of coronavirus, but I'm focused basically in porcine coronavirus. And now I'm going to share you the results of one of our in vivo experiments that we use with neonatal piglets and inoculated them with a different strains of porcine epidemic diarrhea virus. So first of all, actually 7 different coronavirus has been detected in pigs. These viruses are basically distributed in different genera. The most, in general terms, the most severe and also the most worldwide distributed virus are porcine epidemic diarrhea virus and transmissible gastroenteritis virus. Both of them are alpha coronavirus. But the last year, a new species of porcine coronavirus has emerged. For example, the swine enteric coronavirus, the severe acute diarrhea syndrome, and importantly, the Delta coronavirus. That this virus, it's really important in Asia and Europe and in America, but actually there are no cases in Europe yet. Most of these viruses produce a very severe gastrointestinal disease in this model. But there are another species of coronavirus, porcine coronavirus, that can affect extraintestinal tissue, for example, respiratory coronavirus and hemagglutinating and encephalomyelitis virus. The common thing that they have, is that most of them produce a severe gastrointestinal disease, and the reason of that is because the main receptor for most of this virus is the aminopeptidase, which is markedly distributed in the gastrointestinal tissue in both small intestine but also the colonic intestine. And they produce a similar disease, which is characterized by this watery diarrhea, but also we can see vomits and dehydration, emaciation, and quickly death of the infected animals. The most important thing of the virulence and the pathogenicity of these outbreaks is that depends basically of different factors. These factors can be classified of host-dependent factors and viral-dependent factors. The most common and important one are host-dependent, which are the H and interferon 1 and 3 antiviral response in the intestine. But related with the viral strain, this is another important factor, and in cases of porcine epidemic diarrhea virus, it has important. In this virus, in porcine epidemic diarrhea virus, can be classified into different groups depending on the presence or not of the insertions and deletions in the subunit 1 spike protein, and both strains, you can see, are really has a different distribution, but actually this strain is not present in Europe yet. So in order to evaluate the role of these strains in newborn piglets and in the porcine epidemic diarrhea's pathogenicity, we decided to perform this experimental infection where we inoculate suckling piglets, 1 week old piglets, with 3 different strains of porcine epidemic diarrhea virus. One of these strains is an American strain, is a non-Essendale strain, which comes from Iowa University. And the other is an Essendale European strain, it's called Calaf. And we also want to compare what happens with this strain if we previously adapt the virus to the cells and compare with an field strain, which came from an homogenate from the content of the intestine and also the infected mucosa of a small piglet. So this is the experimental design that we performed. We took samples at 0 hours and 48 hours post-infection when the study finished. We performed the necropsy at this time point, and during all this time we observed the animal for clinical signs every 6 hours. So regarding the clinical signs, you can see here the weight evolution from 0 hours until 48 hours post-infection. And you can see that the only animals that showed a mild reduction in the body weight were those animals inoculated with American strain. But the animals inoculated with an homogenated calaf did not show any differences in the body weight. And animals inoculated with the cell-adapted calaf showed a marked increase in the body weight, which were correlated and similar than the negative control. So at 48 hours post-infection, if we compare the weight of the different groups, we saw that most of the animals inoculated with the American strain showed a mild reduction of the body weight compared with the negative control. But we also showed that some animals inoculated with the homogenated calaf also showed a reduction of the body weight, but not of them. But interestingly, there were no differences in the body weight with negative control and the cell-adapted calaf. Regarding the diarrhea, we use this score for evaluate the diarrhea, which ranges from 0 to 3, 0 normal feces and 3 watery feces. And you can see that the diarrhea started at 24 hours post-infection until the end of the study. But the most severe form of the diarrhea was observed in animals inoculated with an American strain. And interestingly, animals inoculated with both strains of calaf did not show differences in the faces, the structure of the faces. And fever, we didn't see fever in any animals, so we assume that these strains did not produce a systemic disease. Okay, So at 48 hours post-infection, we perform the necropsies of all animals, and we use this score in order to analyze the mucosa, the damage of the jejunum and the colonic mucosa, but also we analyze the content of the colon. And this picture that you can see here is the most severe form of the disease, which consists in an animal infected with an American strain. You can see that all the sections of the intestine are markedly dilated. The wall of all of them is really thin, it's like transparent, and the content in all segments is really watery and so with some gas in the colonic segments. So this is a mark, this is a severe diarrhea in piglets. In conclusion with the necropsy findings, we saw that both strains, the American strains, but also the homogeneated calaf, produce a similar damage into the jejunum, the small intestine, but also the large intestine mucosa. But animals inoculated with a cell-adapted calaf did not show lesions in the small and large intestine, and were similar with the negative control. Regarding the content, the same. The animals inoculated with both American and homogeneated calaf shows a marked watery diarrhea in colon, and animals inoculated with a cell-adapted calaf showed more pastry feces in colon, but there were no differences with negative control. Okay, so regarding the histopathology, here you have a normal section of the jejunum of negative control. You can see the ratio between the villi and the cryptal depth. And if we compare negative control with the cell-adapted animals infected with the cell-adapted calaf, we cannot see differences in the mucosa of the small intestine. But obviously, that animals infected with homogeneated calaf and the American strains showed both a marked bilious atrophy and fusion of the small intestine villi. Okay. So in conclusion, we saw that American strain it's the one that produced the more severe bilious atrophy and fusion of the small intestine villi, but homogeneated calaf can also produce a severe bilious atrophy in the small intestine compared with the negative control. And interestingly, animals infected with inoculated with a cell-adapted calaf did not show differences with the negative control. Okay, so in order to detect the presence of virus in this affected tissue, in this affected jejunum, we use immunohistochemistry using this monoclonal antibody from a nucleoprotein of the virus. So it was really interesting because the animals inoculated with the cell-adapted calaf did not show bilious atrophy, but we identified viral particles in these sections. But obviously, both homogeneated calaf, but also the American strain, was distributed diffusely in the apical part of the atrophied villi. So, in conclusion, with the immunohistochemistry, we did not show differences between both strains, American and homogeneated calaf, in jejunal sections. But only the 50% of the animals inoculated with the cell-adapted calaf also had presence of virus in the jejunum sections. And only the American and the homogeneated calaf can be detected in the colonic sections, but we didn't see virus in animals inoculated with the cell-adapted calaf. And all strains can be detected in the mesenteric lymph node, but only the animals inoculated with the American strain has a most consistent result. And finally, we also realized virological study. We performed PCRs using this commercial kit in rectal swabs and jejunal content at 48 hours post-infection. And we can see that both American and homogeneated calaf can replicate similarly in the jejunal segments, But interestingly, it seems that the American strain it's shed higher than the homogeneated calf. And in contrast, animals inoculated with a cell-adapted calf can replicate in the jejunal segments, but it's not shed in feces. And in this case, the viral titer, we use the viral titer, so we performed the viral titer using the qPCRs obtained from the jejunal content, and we saw that the American strain has higher viral load in feces than homogeneated calf, but there are no differences between them. And the viral load in feces of the cell-adapted calf is low, but there are no differences with the negative control. So, with summary, the American strain is the most severe and most pathogenic strain in piglets, according with the results that we obtain in severity, in diarrhea and weight loss, but also with biliary atrophy and fusion, compared with an homogeneated calf. But both strains are similarly distributed in the small intestine and can replicate similarly also in the jejunal segments. But this American strain is shed higher in feces than homogeneated calf, and also it's most infective than homogeneated calf. And finally, the cell-adapted calf did not reproduce the disease because we didn't see loss of body weight, diarrhea, nor biliary atrophy and fusion. But it can be detected in the intestine and also replicate in the jejunum segment of these animals. Okay, so thank you so much for your attention, and also thank you to all my team and the Iowa State University for providing us the American strain and basically the IRTA-CReSA facilities for the in vivo study that we performed there. Okay.
Thank you very much, Carlos. Any questions? Yeah, Dylan?
You need to give Dylan the mic, sorry.
Thanks for that lovely talk. So, did I understand correctly that the major differences between the PEDV strains you used are differences in spike? And what's the nature of those differences? And is it similar to the PRCV-TGV story?
So these strains basically are differentiated by the presence of insertions and deletions in the subunit 1 in cases of S-indel strains that are distributed worldwide. And the non-S-indel indels, I think 200 more or less differences in this segment, and is that insertions and deletions in the subunit 1.
So do you think so? That's the NTD.
Yes, we have identified these differences, but I didn't show you the results.
Okay. And I'm sorry, but do you think that's probably sialic acid binding or something like that?
So, we are writing the paper and we want to publish that. Basically, the result is that all these mutations are affecting basically the subunit 1 is just the place where the subunit 1 and the subunit 2 cleavage, and it's an important place because it's an important place for the pathogenicity of the virus. So we may think that these mutations can be associated with the difference of virulence.
Okay, thank you.
Any other questions? No? Okay, we'll move on to the next talk.
Okay.
Thanks. In the interest of time, people can catch up with Carlos at coffee break, which will be after our next presentation. So it's Tristan Russell from the University College Dublin from Ireland, who's going to talk to us about bovine coronavirus spike sequences for vaccine design. Thanks, Tristan.
Thanks a lot. Hey, so thanks to the organisers for letting me present this work. So I'm going to just jump straight in for the sake of time. So bovine coronavirus is a pneumoenteric virus. This means it causes diseases like winter dysentery and calf diarrhea, as well as being one of the pathogens in the bovine respiratory disease complex. So towards the 5' end of its genome, it encodes predominantly non-structural proteins, which are involved in like replication of the virus and immune evasion, for example. And towards its 3' end is mainly the structural proteins. So these are the actual components that make up the virion, as well as being involved in things like entry. So for this talk, I'm going to focus on these structural proteins. So this is a phylogenetic tree of spike from bovine coronavirus, and I've coloured it based on a location of isolation. So all those in red were isolated from either the Asian or American continents, and all those in green were isolated from Europe. So you can see there's a really clear distinction based on where these natural isolates were isolated. And you can actually see just here, all those to the left are from Europe and all those to the right are from Asia or America. So you'll also see there's this little group of blue isolates. So these are basically really old isolates that are most likely cell culture adapted, or the majority of the vaccine strains. So you can see there's a good bit of divergence between these vaccine strains and the actual clinical isolates that are causing disease. So there is a scour vaccine administered to protect against bovine coronavirus, but only protects against enteric disease, but not respiratory disease. So most of these vaccines are inactivated bovine coronavirus, but recently there was a modified live version also approved for clinical use. So based on this kind of divergence we're seeing between the vaccine strains and the clinical isolates, and in fact there's no vaccine currently for use to protect against respiratory disease, I've got these aims. So the first is to try and predict what are the most immunostimulatory viral proteins. Then I want to see what's driving this variation amongst bovine coronavirus, both within the vaccine strains and the clinical isolates. And I want to see if it's immune evasion that's driving this kind of variation. Then I want to try and update the vaccine so it's more reflective of the clinical isolates and hopefully improve the protection it offers. So this is a bit of a mad slide, but basically all I wanted to show is that I've analysed selective pressures for each of the structural proteins. And I analyse the selective pressures because this is a good way of predicting what proteins might be stimulating the immune response or recognised by the bovine's immune response. So to dig into a bit more detail, I'm going to focus on the spike plot. So I used two algorithms to analyse selective pressures. The first of these was called foobar, and the second of these was called meme. So foobar is represented by these bars in the plot, and this identifies sites under purifying, neutral, or diversifying selection. So purifying means there's a higher than expected rate of synonymous mutations, as in mutations that don't cause an amino acid change. So this would typically occur at, say, like active sites or interaction interfaces, where if you were to get an amino acid change, you'd probably get loss of function of the protein. All the sites under diversifying selection is where there's a higher rate of non-synonymous mutations. So these could occur at immune epitopes, where if you get an amino acid change, it could mean you're able to evade the immune response. Just to summarise those two things. The bars is the results for foobar, and all those annotated residues are the sites that were identified to be under positive selection by meme. So meme doesn't only interest itself really in the diversifying selection. When I looked at all the structural proteins, there's a higher prevalence of diversifying selection in spike and HE, so the hemagglutinin esterase, which suggested to me, or I used to kind of predict these as being the main immunostimulatory proteins for bovine coronavirus. And this is supported by the numbers, just quickly. So you can see in this last column here, for the top two rows, there's a higher prevalence of diversifying, which can also be called positive selection for spike and HE compared to the other structural proteins. There's also a higher amount of purifying or negative selection for spike compared to the others. I might get onto that later. So now I kind of decided to pursue spike and HE for further analysis. And the first thing I did was I predicted the structure of the spike trimer. So I used SwissProt to do this. And what I'm showing here is a view as if you're looking from the outside of the virus down onto it. So you're basically seeing the bit that's exposed on the surface of the virus. And I've just coloured one of the monomers based on the results of the foobar analysis. So each of the residues in green were predicted to be under diversifying selection. All those in blue were predicted to be under purifying, and orange is neutral. And you can see there's a good bit of selective pressure going on for spike. If I was to look into a bit more detail at the RBD, the receptor binding domain, and the selective pressures, so basically what I've done here is I've shown spike from either side, so you can see the front, the back, and the RBD. And I've turned it into a cartoon and shown those residues involved in the RBD as spheres, either here or here. And there seems to be a higher prevalence of selection for these residues in the RBD compared to the residues that are non-RBD, and this is supported by the numbers. So for diversifying selection, almost 11% of RBD residues are under diversifying selection, whereas non-RBD residues is down at just above 5%. And again, there's a slightly higher prevalence of purifying selection for these RBD residues compared to the non-RBD residues. So this kind of makes sense when you think about the function of the RBD. One, it's involved in interacting with the sialic acid receptor, so it has an interaction interface, so changes in this would probably would result in loss of function or loss of this interaction and failure of the virus to enter. But then with regards to diversifying selection, it's recognised by the neutralising antibodies. So there's a pressure to have amino acid changes and evade the neutralising antibodies. So the next thing I looked at was immune epitopes. So I wanted to, So I wanted to kind of rely on some experimental data rather than predict these bioinformatically. So unfortunately, there's been no work done, or I haven't come across any work done for bovine coronavirus trying to actually map the specific immune epitopes. So what I did was I looked at a very closely related virus called human coronavirus OC43. And there was recently in 2022 some work which maps the important residues in the immune epitopes for OC43. And I essentially mapped these onto the bovine coronavirus spike using alignments. And these are annotated as spheres in certain positions. And I've coloured it as on the previous slide. And you can see there is some overlap between these sites predicted to be under diversifying selection and the predicted immune epitopes. And this is supported by the numbers. So again, there's a much higher prevalence of diversifying selection at these residues that are predicted to be immune epitopes. There's also a higher prevalence of purifying selection, which is a bit surprising. Haven't really got an explanation for this yet, unfortunately. So I also looked at HE. So the structure for hemagglutinin esterases has been resolved by X-ray crystallography, and this is just the surface that I'm showing here with the same annotations or the same colours as before. And again, for the RBD, I turned the whole protein into a cartoon, but then made the RBD residues into spheres, and then coloured them as before. And again, you can see there seems to be a higher prevalence of diversifying selection within the RBD, again suggesting it could be recognised by the immune response. And this is supported by the numbers. There was about almost 5% of residues in the RBD under diversifying selection, whereas around about 4% for non-RBD. There's actually a lower prevalence of purifying selection for the RBD residues, which goes against what I said with spike, but when you consider HE as an enzyme and outside of its RBD binding domain, it has catalytic activity. This kind of makes sense then because a lot of these residues are involved in the esterase activity of HE are likely to be under purifying selection. So now I'm going to go on to a bit of the sequencing and surveillance I've done. So I received isolates from Department of Agriculture, Food and Marine in Ireland, and I sequenced, first I PCR amplified my genes of interest, spike and HE, and then I Sanger sequenced them. So you can see for spike, compared to the Mebus strain used to design the vaccine, there was 49 amino acid changes in my Irish isolates. When I did some phylogenetic analysis of these spike sequences, you can see they fall into 1 or 2 clades, shown in blue and green here. When I dig down into these clades, there was actually a bit of diversification, or a bit of kind of divergence, based on location. So the majority of clade 1 were from northern and western counties, and a majority of clade 2 were from, or all of clade 2 sequences were from the southern and eastern counties. So this could just be a coincidence. All I'm actually seeing is 2 introductions of bovine coronavirus into Ireland, or it could be that there's some kind of environmental factor associated with a location that's driving this divergence in spike. But it would just be for spike because I don't see the same pattern for HE. So the majority of the sequences fall into 1 or 2 subclades, shown in pink here, but there's no real any grouping based on location. There was 1 real random sample 117 from Offaly, and when I did a blast search to see what was the closest sequences, it was actually a bovine coronavirus isolate taken from an oryx in Israel. No explanation for this, but it was just a bit random. And in total, there was 10 amino acid changes compared to the Mebus sequence, which is about almost 2.5% of the residues. So to go back to the structures, I mapped all these sites with amino acid changes onto the structures I have for spike and HE. So this is the stalk, this is the kind of surface exposed part of it, and this is HE. And I showed the residues with changes as spheres, and then the rest of it as cartoons. And obviously, well, not obviously, but as expected, there's a good bit of overlap between sites that were predicted to be under diversifying selection, so those in green, and where these amino acid changes occur. And also there's a lower prevalence of purifying selection at these sites where there's amino acid changes. So the next thing I did was I looked at whether there's a good bit of overlap or whether there was any overlap between the predicted immune epitopes and the sites with amino acid changes. So again, they're shown as spheres for the amino acid changes and shown in pink for the predicted immune epitopes. And there is some overlap. And actually, when I look at the numbers, there's a much higher prevalence of amino acid changes for these residues, which are predicted to be immune epitopes, suggesting that the changes that are occurring are at least in part driven by a pressure to evade the immune response. So in summary, I've shown there's a good bit of divergence between the vaccine and the clinical isolates. And then I did some selection analysis on all of the structural proteins, and so there's increased levels of diversifying selection in spike and HE. And then there's also a good bit of overlap between these sites under diversifying selection and the RBDs, and in the case of spike, also purifying selection. I did some sequencing and showed there's a lot of variation, or a good number of amino acid changes between these Irish isolates and Mebus, the strain used to design the vaccine. And there's a good bit of overlap, as expected, between diversifying selection and amino acid changes. And there's also a decent bit of overlap between these amino acid changes and the predicted immune epitopes. So to go on to the final, this is just one slide, don't worry. The final kind of bit of my, the aim was to update the bovine coronavirus vaccine. So to do this, I want to use the sequences I have for spike and HE and design an mRNA vaccine, which I'm going to do in collaboration with a Belfast-based company called Fion, and have these so they're more representative of the clinical isolates that are causing disease in Ireland. So I'm going to steal or use a lot of the kind of modifications, sorry, my earpod, a lot of the modifications that have been shown to work for COVID-19 vaccines with my bovine coronavirus vaccine, such as introducing a diproline mutation that locks it in its pre-fusion conformation, as well as using, say, pseudo-uracil instead of the normal uridine, as this kind of reduces the innate immune response and the inflammatory response, which reduces the cytotoxic side effects. So I've already designed this, have the sequence and everything. It's just a case of actually generating it. Then once I've generated it in collaboration with Fion, we're also going to test it in mouse models to check it induces a good immune response. Then, depending on how this goes, potentially progress it into cattle. So I'd like to thank all my funders. So that was DAFM, the Department of Agriculture, Food and Marine in Ireland, my lab, especially Gerard Barry, and Kaylin, who kind of got all the PCRs for the sequencing up and going. Emma is doing some surveillance at the moment of viruses in slurry, which might be of interest to people. I'd like to thank Jose Maria Lozano, who supplied all the samples from Back Western Laboratories in DAFM, and hopefully my future collaborators, Helen and Lindsay from Fion. So thanks a lot.
Tristan, have you got a quick question before we break for coffee? I've got one for you. So your two clades of wild-type viruses that you've got different parts of Ireland, so are they from different breeds or different husbandry? I
haven't got information on breeds, unfortunately. I do have information on the things like how badly the disease was and stuff like that. Like what was the actual disease presentation, but there was no kind of links in that sense. It's pure, the only pattern I could see was based on location.
Okay, so it might be interesting to have a bit more epidemiological data to kind of go with that.
Yeah.
Okay, great. Thanks very much. I'd like to thank all of our speakers from our first session. We'll break for coffee until just before 11 when we'll start the second part of this session. And I would like to remind people, if you're not a member of UKICN, then you can sign up. It's free. There's a QR code out on the poster that we have. And we really encourage everybody who's attending today and also people in all of your labs to join the membership, because then you get updates and notifications of future meetings, which includes our AGM, which will be held in September in Slovenia, and we hope to see many of you there. Thanks. Back for 11 o'clock, please. Thank you.